This audio lecture is designed to accompany Chapter 12, and this chapter covers many aspects of mutation. You'll notice that in the Chapter 3 folder, I've actually put together a little review of protein synthesis, so transcription and translation. I think it's good to kind of take a look at that to remind yourself how protein synthesis works. Specifically translation. So how the information in DNA that is then transcribed into an mRNA strip is ultimately interpreted in the cytoplasm as a series of amino acids. And those amino acids are the building blocks of proteins, because we're going to see that mutations, although not always, can cause changes to proteins based on alterations to that strip of information and the amino acids associated with it. A mutation, purely defined, is just a permanent change in a DNA sequence. That's all it means. So you'll notice that there's no negative connotation to this definition, right? It's just a change, simply a change in a DNA sequence. So from an evolutionary perspective, mutation is extremely important, and that's because it generates genetic variation. Without this genetic variation, we wouldn't see variations in phenotypes. And the variation in phenotypes, especially with regards to living organisms, is really important because those phenotypes can sometimes be associated with increased survival or reproduction. And so that's how we see evolution over time. It starts as simple mutations, sometimes even just a single change in a DNA sequence. And as long as that change is associated with positive benefits, like increased survival, increased reproduction in the face of environmental change over time, that mutation is going to become increasingly present in the next generation. So over time, within the population, we can see changes. So that's all mutations are. They're simply a change. A mutation at the very smallest scale, so the simplest change that we can make in a DNA sequence, is in a single nitrogenous base. So remember that the building blocks of our DNA, or the monomers of DNA, are called nucleotides. And those building blocks themselves are made up of 3 repeating units. We have our sugar, which, because we're dealing with deoxyribose, nucleic acid is deoxyribose. We have a phosphate group. These are the repeating units that we see over and over again on the outside edges of our ladder, or double helix. And then we have our varying component, our nitrogenous bases. And this is what can be different here, right? Because we have 4 possibilities. We have our adenine and our guanine, which are our purines. And we have our cytosine and thymine, which are our pyrimidines. So when we're dealing with just a simple change to a single base, we call that a point mutation. And we can look at this nice sketch that I have included here. So we have our original DNA sequence on the top. And let's get this directionality here. So we're going to say this is our 5' to 3' end. And what we see that in our original sequence, at the 5th nitrogenous base position, we have a thymine. But due to an error during replication, that thymine changes to a cytosine. So that means during replication, as the the strand of DNA is actually being constructed by DNA polymerase, rather than putting in a thymine, it accidentally inserts, as we see here, a cytosine. So that's a mutation. And if that mistake is not caught, because remember, DNA polymerase can also proofread and check for mistakes. But if it doesn't notice this mistake and that mutation sticks around, it's a simple change. It's called a point mutation. On a larger scale, mutations can involve entire genes, portions or sections of a chromosome, and even an entire chromosome. So we can see in this image, we've got a total structural modification of a chromosome. So an entire portion of the arm is removed. That's a mutation. We may also see copies of entire chromosomes. So we've already learned about non-disjunction. Remember, that is a mistake that happens during meiosis when the homologous chromosomes are being separated during anaphase 1 of meiosis, or sister chromatid copies are being separated during anaphase 2 of meiosis. During either of those times, if those things fail to separate, that's called non-disjunction. And when that happens, we end up with extra copies. Can't fit my N on there, but you get the picture of chromosomes. And we're going to learn more about entire chromosome mutations in chapter 13. Mutations are actually pretty rare, and that's because DNA polymerase has the ability to proofread. So during replication, if DNA polymerase makes an error, it also proofreads, checks for errors, and can fix them. But it's not perfect every time. And that's how we end up with these permanent mutations. In terms of how many there actually are, scientists have looked at these events and have determined that in humans, there are about 10 to the negative 10th mutations per base pair. So thinking about all of the base pairs in our genome per replication event. So that value turns into thinking about the fact that our human genome contains 3 times 10 to the 9th total base pairs. There are about 10 to 100 mutations per genome per generation. So we'll talk about why this is such a big range, 10 to 100, on the next slide. But what I'm referring to here when we're talking about per generation, at least on this slide, when we're passing on these mutations to the next generation of offspring or individuals, we call this a germline mutation. So these are mutations that are occurring during replication before meiosis. Right? The process that creates the nuclei of our gametes, our sperm and egg. And so because these mutations are in the genome that are going to be passed on to the next generation of individuals, we call these germline mutations. So make sure that when you're reading the questions on the quizzes or exams, I'm talking about generation, that you're paying attention to the generation. Does the question ask about individuals, or does it ask about generation of cells? So in terms of these germline mutations, what this means is that from the start of you becoming a zygote, so after sperm and egg combined, you were a zygote, through the production of all of your gametes, your genome, at least as it's being passed on from your spermatogonia or oogonia, those precursors, those stem cells that hang out in our gonads, the testes and ovaries, to the time that we create our viable sperm, viable eggs, that genome has accumulated 10 to 100 mutations. The reason we have this range is that men actually represent the higher end of that value. So men actually have more chances of passing on mutations, at least within the genome. And that's because, remember that ultimately they're continuously producing their gametes. Sperm production continues for a long time, and they produce many, many more sperm than eggs over the course of even just thinking about a month. So just one cycle that a female goes through, males can be producing millions of sperm. So many more rounds of meiosis, which means many more rounds of replication that preceded meiosis. So a greater chance of mutations, or the greater the number of mutations. So I just want to point out here, as I mentioned on the previous slide, that we also have mutations that can be passed on to our somatic cells, right? So thinking about our somatic cells, so all of the other cells in our bodies besides our gametes, they obviously can end up with mutations as well. So that's in the next generation of cells. So somatic mutations happen the same way because we've got DNA replication that precedes mitosis, right? So our cells are going to pass on their genome to the next generation of cells. So we need replication to occur. If there's an error and DNA polymerase doesn't catch it, that mutation is passed on to the next generation of cells. This mutation rate, so the somatic mutation rate, really depends on the type of cell. So some of our cells, like our skin cells, are very frequently undergoing mitosis, right? So we have a need to replace or replenish those cells in areas of our body that see continuous use, like our skin, like the lining of our esophagus, the lining of our intestinal tract. So there's lots of replication, lots of mitosis. So those cells are more likely to end up with mutations versus cells in our body that tend to divide more rarely. So like our neurons in the brain or our liver cells. So these mutations are passed on to the next generation of cells in our bodies, but not transmitted to the next generation of individuals. So just a thing to keep track of. Mutations can also occur in any part of the genome. Remember that our genome is made up of lots of different components. Everybody talks about genes, right? The genes that influence phenotypes, but we've learned that that is actually a small proportion of our genome. So mutations can occur within the introns of genes. So remember, those are the non-coding regions that are actually removed the very end of transcription before the mRNA transcript leaves the nucleus to the cytoplasm. Mutations can occur within the exons. So the portions of the genes that are ultimately going to be part of the mRNA transcript that then will be interpreted as a series of amino acids and ultimately a protein. Mutations can occur within our genes that control processes. So that's a huge portion of our genome, right? Most of the genes that we have actually, rather than encoding proteins for things that we see as phenotypes, they control processes within our bodies. So whether that be replication or they help with mitosis, meiosis, transcription, translation, we can see mutations there. We have those telomeres, so the long stretches of repeats at the end of our chromosomes that protect them during rounds of division, or other stretches of repeated nucleotides throughout the genome. And we also have, remember, mitochondrial DNA. So our mitochondria have their own little circular DNA, and that DNA is being passed on to the next generation of mitochondria and ultimately to offspring. So mutations can occur in any of these regions. However, the mutation likelihood and the rate among these different regions varies. And we're going to talk about the reason behind this on the next slide. In some cases, we understand why the mutation likelihood and rate varies depending on the location within the genome. So one great example is that the regions of our genome that have long stretches of repeated bases. So either in our telomeres or just other regions within our genome. Those long stretches of repeated bases are way more likely to end up with errors during replication. We've actually talked about this already when we studied replication in chapter 9. So those areas where we have many, many repeats, we can easily see mistakes, and that's because it's really difficult in those regions for DNA polymerase to keep track of where it is. It makes sense if you were to be typing out yourself, you had a long series of, let's say, ATs, that you were typing up from looking at your textbook and typing it onto your computer. It's really difficult to keep track of how many that you're placing in order. And it's the same thing for DNA polymerase. It's just easier for mistakes to be made. Now, those mistakes in regions of these repeats often don't have any consequence because those stretches of DNA often, they aren't encoding proteins or proteins that are important in any processes. But we can make use of this information or take advantage of this because these repeats then often vary between individuals and even sometimes populations. So as a wildlife biologist, I use this information to study how individuals move across the landscape or what populations they come from. And we term these things microsatellites. So they're just regions within the genome that have a higher likelihood of having these mutations. So just individuals tend to be different from one another with regards to how many mutations they have and what those mutations are, so we can keep track of them and compare them across populations. Mitochondrial DNA is another great example. We know that it mutates at a much higher rate compared to just nuclear DNA in general, all nuclear DNA. And that's due to the fact that there aren't really any repair mechanisms for when there are changes, and the fact that mitochondria are constantly being exposed to free radicals. And we've already talked about these differences between mitochondrial and nuclear DNA. But because mitochondrial DNA also mutates, it's a way for us to kind of study those changes. And it's very often that scientists that are looking at population movement in terms of humans, looking at how individuals have moved across our landscape over time, like broader timelines over geologic history. The next point we're going to cover is the fact that the effects of mutations vary. So what does that mean? Not all mutations are bad. It's a huge misconception in the field of genetics from the general public's perspective that when you see the term mutation, it evokes a negative connotation, right? We think of mutants as being bad. But in actuality, the effects of mutations vary. A mutation can have absolutely no effect at all. A mutation can have a positive effect, right? Those are the mutations that offer some type of survival or reproductive advantage, which can then drive evolution in the face of change. Mutations can also have a negative effect. So the one that kind of general public thinks of most often as negative effects, yes, that happens too. But interestingly, most mutations are actually neutral, or at least nearly neutral, when you think about the large scale of possibilities here. So most mutations actually have no effect. And that can either be because they are within a region that doesn't include a protein encoding gene, right? So if you have a mutation within this long stretch of repeats, doesn't really matter because that stretch of DNA isn't encoding for an important protein. But we're also going to learn, because of the unique aspect of the genetic code. Remember that the genetic code, when thinking about our genetic code, we have, in many cases, multiple codons that code for the same amino acid, right? So we have that redundancy built into our genetic code. And because of that, we can sometimes have mutations that don't have any effect simply because it changes the codon such that the same amino acid is going to be present, whether the mutation was there or whether it was not. So here I'm just showing you a figure from scientific literature. So this was published in textbook Hedrick in 2005, and it just shows you how we've got frequency of mutations that are plotted here. And it's showing us, when thinking about all of our mutations, and on our x-axis, we're dealing with fitness. So fitness is just how these mutations are affecting the individual. We've got kind of negative effects on one end. So those are lethals, and then other kind of negative consequences of our mutations. And then we've got positive mutations on this end. So we can see that those are more rare than the bulk of mutations, which fall in the intermediate range of being neutral and nearly neutral. I mentioned this on the previous slide when thinking about these long sections of repeats, how these neutral mutations are really useful because we see mutations pop up. They don't have an effect, so they're neutral, but we can take advantage of that and use the information to measure gene flow. Gene flow is simply the movement of genes across the landscape, so the movement of individuals across the landscape. And genetic drift just studies the changes of the genetic information within a population over time. So we can use these neutral mutations that have no effects, that are the most common, the things that we see, to study different population processes over time. So what about when mutations do alter the structure and function of genes? So let's say we have a protein encoding gene, and a mutation actually has a consequence. What happens? Well, we've already covered this a bit when we discussed Mendelian genetics in chapter 4. So we have a change that can cause a loss of function. And so what that means is those mutations reduce or completely eliminate the gene of interest product. And those are, or at least tend to be, recessive, right? So those are our recessive alleles. We can also have mutations that cause a gain of function. So it changes a gene's product and activity. And those mutations tend to be dominant. So we're going to explore how these recessive and dominant mutations can cause disorders later on in the lecture. Here on this slide, I'm just revisiting the idea that when talking about mutations, you need to keep track of where the mutations are occurring. So germline mutations are changes that occur during DNA replication that's going to precede meiosis. Because of that, any changes are going to be passed on or transmitted to the next generation of individuals. Those are the mutations that we follow over time across a pedigree. We also have somatic mutations. So those are changes that occur during DNA replication that then precede mitosis. So ultimately, changes in the genome that are being passed on to the next generation of cells. So what causes these mutations? Well, mutations can either occur spontaneously, or they can result from exposure to what we call a mutagen. So that would be a chemical or radiation, UV light, things that are available in the environment, so external factors that can trigger mutations. So in this lecture, I'm going to focus on spontaneous mutations. So spontaneous mutations are new. We can also refer to them as de novo. So that means we've got a mutation that shows up and kind of can surprise us, right? So you've got parents that end up giving birth to a child who has some type of condition or different phenotype than expected from family history. And that's due to this de novo or new mutation. And these types of mutations, I've already mentioned, arise due to errors that occur during either DNA replication, right? So DNA polymerase is making a mistake, and that mistake is being ultimately passed on, or during meiosis. And so those tend to be larger scale mistakes and involving either portions of chromosomes or entire chromosomes. And so that is actually going to be covered more in depth in chapter 13. This chapter covers errors that occur during DNA replication and what those errors entail. What I really like to focus on from this chapter in terms of quiz questions, exam questions, are looking at these different types of mutations that we see and what their effects are. So we've already talked about the fact that there can be many different types of mutations. And these mutations are simply classified by how they affect DNA structure or how they affect the function of the gene product. So keep in mind that we've got mutations that can occur on the smaller scale here, so just a single base change, versus mutations that occur at larger scales, so whole portions of genes, for instance. So this is all covered in section 12.5 of the chapter. So we've got a whole section on types of mutations, defining them, and their consequences. And I really like this table that's on page 215 of your text. So when we think about our genetic code, we're thinking about a protein encoding gene in our DNA. And we've talked about how our DNA, when it's coding for a particular protein, it's because the sequence of bases, the sequence of letters within the DNA are ultimately encoding a protein product or a sentence that's being read, right? So the letters themselves occur as codons or units, which would be our words, and then are ordered together to produce this complete sentence. So this table shows us when we've got our normal DNA sequence, we would want, thinking in our analogy of a protein to a sentence, we should end up with the one big fly had one red eye. And then we can see in each of these different mutations how a change in the lettering can either alter a single word within our sentence, can alter many words within our sentence. So we can lose part of the information. Looking at our frame shift mutation, we see things that just ultimately end up looking like gibberish. We can't even read the rest of the sentence. So on and so forth. I think this is a really great way to help your brain kind of wrap around what these mutations do in terms of functionality of the proteins. On the next several slides, I'm going to walk through each of these different mutations listed in table 12.4. I'm going to define them for you, show you how DNA structure is altered, and then talk about the consequences for function. So we're going to start with the easiest mutation, the simplest mutation, and that's our point mutation. So remember, that's just a change in a single base. So we have our DNA sequence, and we see one base is substituted for another. We can further classify a point mutation as either a transition or a transversion. And the difference between these just depends on what our original base was and what we're changing it to. So transitions are actually more common. And this is when we have a purine replacing a purine or a pyrimidine replacing a pyrimidine. So remember that our bases fall into two different categories, purines and pyrimidines. Our purines are adenine and guanine. Weirdly, I just remember that as ag, like agriculture is pure. So purine, A and G, and pyrimidines are cytosine and thymine. So if we look in this top example here, a purine is replaced by a purine. So transitions could be where we originally had an adenine, now we see a guanine, or originally we had a guanine and we see an adenine. Or we may have the case as we see in the image above, where the original sequence had a pyrimidine. In this case, we have a thymine. And then we have a mutation where we have a point mutation, a change in a single base is substituted. In this case, it's a cytosine. So we're replacing the initial base with the base that has the same form, in this case, cytosine. And because that replacement is made, we call it a transition. Point mutations can also be described as transversions. And this happens when we have a base that is structurally in a different class than the one that we started with. This can happen in two ways. We can have a pyrimidine replace a purine, or a purine replacing a pyrimidine. So we've got two tables showing how this can work. In this first table, originally our base was of the purine class. And then we have a mutation that changes the purine to a pyrimidine. And the bottom table is just the reverse of this. So we can start with a pyrimidine and end up with a purine. So because we're changing a base to a completely different class, a different carbon ringed structure, this is called a transversion.